Biology 2121, Presentation 11, Nervous Anatomy. Our current presentation is going to dwell primarily on the central nervous system, and an adequate understanding of the central nervous system needs to begin with an understanding of its embryonic origins. So I remember from our discussion a while back on the embryonic germ layers found in humans, ectoderm, mesoderm, and endoderm, we learned once upon a time that ectoderm is ultimately that embryonic tissue from which our central nervous system is derived. So let's take a look at the process. I noticed that in the embryo, and by the way, these illustrations that I see, illustration 1, 2, and 3, going from left to right, represent different steps in the development of what's going on a dorsal aspect of a developing human embryo. And so what I see is a little pen and ink drawing showing me a cross-sectional plane indicating to me that I'm looking at just the dorsal aspect of that particular developing embryo in cross-section. And I see purple, which is ectoderm, and I see some peach-colored material, which is mesoderm, and the dark orange cells constitute endoderm. And by the way, the yellow stuff is really a space. It constitutes the body cavity or the coelom. But you'll notice what's going on is at the dorsal aspect of the embryo, a neural fold is starting to occur. That is a groove that is oriented lengthwise along our embryo. And it's invaginating, it's going in, it's going in, it's going in, and at some point at the surface, convergence is going to happen, and the superficial ectoderm will meet, and at that point, the invaginated ectoderm becomes isolated as a neural tube. If you take a look at our illustrations, starting with the illustration at left, you'll see the neural fold forming with its neural groove. You'll see the convergence taking place at the dorsal aspect. You'll see the superficial layers of ectoderm join. You will see at that point the neural fold isolated as a neural tube, and you'll notice there are some additional cells scattered to the right and the left of that neural tube. Those are identified as the neural crest. The neural crest are a group of stem cells that can migrate laterally and will eventually contribute to the formation of the peripheral nervous system. While my description is certainly adequate, please do not discount the text and the bold print terms that are on this slide. And you will see the same illustration in your anatomy and physiology text as well. I must say that this particular slide, primary and secondary vesicles, is going to be critical to your success on a number of questions found on the exam. So let's pick up where we left off. On our previous slide, we mentioned the fact that we form a neural tube. And the slide specifies that the anterior portion of that neural tube becomes the brain. So we're now going to take a look at what happens to that neural tube as we move towards weeks 3, 4, and 5 of embryonic development. As the weeks go by, the anterior portion of the neural tube becomes thicker and becomes more developed, at which point we see a regionalization going on. So week 4 post-conception, you will need to know there are 3 different regions or sections to the anterior portion of our neural tube. These are referred to as primary vesicles. They're called vesicles because technically the brain is hollow, because you remember this is a neural tube, so there's an opening smack dab in the middle. So I see one vesicle labeled as a prosencephalon, also known as the forebrain. I see a second section illustrated in yellow labeled as the mesencephalon, and I see a more posterior region in red labeled as the rhombencephalon. Prosencephalon, forebrain, as I mentioned before. The mesencephalon is the midbrain, and the rhombencephalon is the hindbrain. What is occurring after or posterior to the rhombencephalon is going to ultimately become the spinal cord. Now, let's take a look at what happens between week 4 and week 5. See, this is one of the more critical periods of time in embryonic development where it's really hit or miss as far as the central nervous system is concerned. Notice that the prosencephalon itself becomes derived into something called the telencephalon that eventually becomes the cerebrum, something called the diencephalon, which has some very, very critical brain structures as well. I notice that the mesencephalon stays the mesencephalon and is still considered to be the midbrain. But then look at the rhombencephalon. It is derived into two regions itself. One called the metencephalon, which becomes the pons and the cerebellum in the mature brain, and then the myelencephalon, which ultimately becomes the medulla oblongata, or the brainstem, in the mature human. So at this point, we have 5 secondary brain vesicles: the telencephalon, the diencephalon, the mesencephalon, the metencephalon, and the myelencephalon, all being derived from 3 original primary vesicles. Now, if you take a look at the lateral views, that's kind of helpful. The lateral view shows me that as formation takes place, about week 5, the central nervous system starts kind of coiling into kind of a question mark shape. And that's going to be critical because when we look at the fully formed brain, the conspicuous part obviously is the cerebrum, but buried in the cerebrum is where this diencephalon and the mesencephalon are going to be found. And then you got the rhombencephalon, which ultimately becomes the cerebellum and the medulla. This fact will become more obvious when we see our next slide. Here is our illustration, labeled illustration, of the major parts of the mature human brain. And before I forget, let me just go ahead and clue you in at that last comment I made on the previous slide. I said at about 5 weeks, the central nervous system, that is the anterior portion of the neural tube, starts to kind of coil into a question mark shape. And I said that the major conspicuous part of the brain is the cerebrum. And so there I see it. The cerebrum on the right-hand illustration is derived from the telencephalon. Remember, the telencephalon is part, ultimately, of the prosencephalon. But then buried inside of that, and it's kind of highlighted with a little square, and it's actually illustrated in greater detail, I see the diencephalon consisting of the thalamus and hypothalamus, and immediately below that, I see something labeled as midbrain. That would be constituting those next 2 sections of the 5-week brain, the diencephalon, and then the mesencephalon being the midbrain. But then you'll see coming off of that would be the pons and the medulla and the cerebellum. So the brain is essentially kind of like a question mark in terms of its organization. The conspicuous part being the cerebrum. Then you notice the cerebellum and the spinal cord derived from the posterior region of the neural tube and the rhombencephalon, and then smack dab in the middle would be where I find the diencephalon, and the mesencephalon is part of that question mark shape. So now you'll need to know on this illustration is the cerebrum. You'll notice that the cerebrum consists of fissures that are referred to as sulci and ridges called gyri. We'll talk about them on another slide. You will notice the position of the cerebellum. You will notice the relative positions of the diencephalon, the thalamus, the hypothalamus, and the midbrain, the pons, and the medulla oblongata, which ultimately connects to the spinal cord. And we will review this again and again and again. It will continually come up over the next series of slides. But make sure you are aware of these major parts of the brain, and make sure you're aware of what they are derived from. They're derived from 5 secondary vesicles, and you'll want to know those 5 secondary vesicles are derived from those 3 primary vesicles that started off way, way back about 3 to 4 weeks post-conception. The following 4 or 5 slides will focus on the cerebrum, obviously the largest of the 3 major regions of the brain, but the cerebrum really controls a significant amount of sensory information and a good degree of motor information as well. Cerebrum is where we develop conscious thought. Important, and probably I would have to say the most characteristically human portion of our brain. As you move from the cerebrum through the midbrain to the hindbrain, our brain becomes much more concerned with more vital and I would have to say more primitive aspects of control. Vital control, well, all vertebrates have that. All vertebrates have respiration. All vertebrates control heart rate. But when it comes down to it, thought, emotions, higher order processing of information, that's a distinctly human thing. So let's take a closer look at our cerebrum. We already mentioned some general characteristics. Now I'm looking at it a little bit differently. I see we have 2 halves of the cerebrum, the right and the left cerebral hemispheres, and they're characterized by the presence of those sulci and gyri I mentioned not too long ago. The surface of the cerebrum consists mostly of gray matter, is what we refer to as the cerebral cortex. More about that in a minute. And I noticed that smack dab down the middle of the brain, there's something called the longitudinal fissure, which is some big cleft separating the right and the left cerebral hemispheres. But deep in the middle of all that, I see indicated in a dotted line something called the corpus callosum. You've probably seen the corpus callosum a lot better in laboratory. And if you want to revisualize it, please Google corpus callosum, because this is not a really good indication of its position and certainly what it does. Because the corpus callosum is a place where the right and the left hemispheres interconnect with one another. The corpus callosum is composed largely of white matter. And white matter is characterized by neurons with myelinated axons. And the idea is that if I am going to have some integration of processing going on in the right versus left cerebral hemispheres, I would want those to interconnect with one another and also interconnect with what's going on a little bit further down the brain. Ultimately, all of that information is going to have routed into my brainstem and down my spinal cord, ultimately to my peripheral nervous system as well. You are aware, I'm sure from laboratory, that the cerebral cortex is divided into a number of different regions, what we call lobes. And the gyri, which are the folds of gray matter, are one way that the cerebrum increases its connectivity and also increases its delivery of nutrients to all of these neurons that are both oxygen needy and nutrient needy. And they're critical. And so increasing surface area will go a great way to providing them with the oxygen and the nutrients they need. And then you've got these grooves. And the grooves actually not only are present between the gyri, but also are present in making distinctions between these lobes. And so those major sulci are sometimes also referred to as fissures. We mentioned the longitudinal fissure that divides the cerebral cortex into right and left cerebral hemispheres, but then I see sulci dividing the cerebral hemispheres into their constituent lobes. We identified the lobes in laboratory: the frontal lobe, the parietal lobes, the temporal lobes, the occipital lobe, and these sulci dividing those lobes as follows: the central sulcus, separating the frontal lobe from the parietal lobe, the parieto-occipital sulcus, which separates the parietal lobes from the occipital lobe, and then finally the lateral sulcus, which separates the parietal lobes and frontal lobes from the temporal lobes. Now that we've got a handle on some of the general anatomical characteristics of our central nervous system, at least the brain, we understand that we've got the cerebrum, we've got the cerebellum, we've got the medulla oblongata, also called the brainstem. We know they all have some degree of embryological origin from the original primary vesicles giving rise to the secondary vesicles, and we understand some of the anatomical characteristics of the brain of the mature individual. But now, at this point, what we're going to do is we're going to try to connect anatomy with function. And in order to do that, we are going to begin with a very brief discussion about a fellow by the name of Brodmann. Korbinian Brodmann, or more appropriate pronounced Korbinian Brodmann, was a German neurologist. In the early 1900s, did a very, very significant amount of work on the anatomical organization of the brain. He took a look at the microscopic structure of different areas of the cerebrum, and in doing so, he had to make thin microscopic slices, stain those slices, and study the neurons and see how those neurons were organized. To make a long story short, after many, many, many years of work, he identified 52 distinct anatomical regions of the cerebral cortex. And those areas were based on the neurological structures present. That is the organization of the neurons. Now, he didn't specifically connect these with specific functions, but as the years went by, neurologists studied the brain in terms of where functions were located in terms of the cerebral cortex, and they found that these areas identified by Brodmann, nowadays known as Brodmann's areas, corresponded quite well with many of the functions of the cerebral cortex. So our illustration here are actually Brodmann's original illustrations, and the publishers have superimposed some colored areas over them. And you'll see some names, and you'll see some indications of where these locations are. Now, our next slide is going to be a much better depiction of these areas, and there are going to be some areas you're going to need to be aware of in terms of not just what they're called and where they're generally located, but more importantly, you're going to need to know something about their function. So let's take a look at that on our next slide. Okay, so we've got a much nicer looking illustration here, and the title of our slide reads, Cerebral Cortex Contralateral Control. Left controls right, right controls left. So before we actually talk about the mapping of some of these functional areas of the cerebral cortex, remember you've got a left cerebral hemisphere and you got a right cerebral hemisphere. And you understand both of those cerebral hemispheres are connected by something called the corpus callosum, which is a region of white matter that is buried deep underneath of the cerebral cortex. And the corpus callosum is a switching center where we can take impulses from the right, move them to the left, move them from the left, move them to the right, and interconnect with the spinal cord that ultimately is going to carry this information to motor effectors in the body. But the fact of the matter is what's going on in the right cerebral hemisphere, as a rule, controls motor effectors on the left side of the body, and vice versa. Hence, left controls right, and right controls left. But now let's take a look at the functional areas. And once again, I will mention these areas correspond quite well with Brodmann's original designation of areas based on his cytoarchitectural work in the very early portion of the 1900s. But I'm going to need you to know the names. I'm going to need you to know where they're located. And more specifically, I need you to know what they do. And I will be very careful to make sure that you know exactly which ones on this slide you need to know. In fact, I think probably what the best thing for me to do is, rather than run down the list right now, I am going to go ahead and stop recording this lecture at the moment, get on Word, and generate a table listing all of these areas that will give you an opportunity to fill in where they're located and what they do. And that way you get a chance to actually do all this, put it in one nice tabular form that you can use to study from, but also the actual doing, the actual of looking up what is the premotor cortex, where is it located, and what it does is going to go a great way towards helping to reinforce this information. And like, once again, it's going to be something that will, I promise you, show up on exam number 4. Okay, so once again, from our last slide, there are certain areas of the cerebral cortex you're going to need to know by name, where they're located, and what they do. I posted a chart to fill in for you. It's in the student information folder on D2L, and you're more than welcome to download it and print it out and fill it in. And this particular slide, it's titled Cerebral Cortex Damage, talks about some of the issues that can arise when certain cortical areas, and the word cortical implies cortex, certain cortical areas might be damaged, and there's going to be some practical connection between the results of damage and the actual functions that you identified in the previous slide. Please be sure of this, particularly with regard to Broca's and Wernicke's area concerning speech and interpretation of speech. That content will find its way on a question or 2, or perhaps 3, on exam number 4. So be prepared. Okay, our present slide, basal nuclei act together to form motor pathways, is important from the standpoint that it's going to allow us to understand exactly how we integrate all this information from our cerebral cortex. But the thing is I'm not going to ask you specifics on this slide. So this is what I want you to understand. It is important. We've got a lot of sensory information coming in, and that sensory information, for the most part, is routed through a part of our brain called the thalamus, and we'll talk about that in a minute. And the thalamus routes all that information to the various areas of our cerebral cortex. Well, you think about it, anything you're doing probably involves a number of different processing events that are going to be taking place all over your cerebral cortex, and it's all got to be integrated. And once we react to that, or once we process the information and we're generating motor impulses that are going to be traveling out efferent nerves, those impulses need to be routed to the appropriate efferent pathways. I mean, you don't want information traveling to your big toe if you really need it to be traveling to a bicep muscle. So fact of the matter is all of this information being generated by the cerebral cortex has to be integrated somewhere. And what does that are regions of cell bodies located in the brain called the basal nuclei. And you'll see them illustrated here in various shades of dark and blue, and you notice that they're located more or less at the base of the cerebrum, deep inside the cerebrum, right down about where our corpus callosum is. And so these basal nuclei are groups of cell bodies that are going to be gathering all this information from the various parts of the cerebral cortex and then making sure they are sent out to the appropriate effector pathways. Now, some of those effector pathways are going to be excitatory. They're going to ramp up activity, and some of them might be inhibitory. They're going to ramp down activity. But it's the basal nuclei that go by a number of very intense specific names that I'm not need you to worry about, are going to be sending that information to the appropriate effectors out there amongst the peripheral nervous system. Okay, more about basal nuclei, but the good thing is more about what you don't need to know. On the previous slide, I mentioned there were these basal nuclei that were responsible for routing impulses from the cerebral cortex to the appropriate motor pathways. Now, those basal nuclei go by a number of rather intense names. They were mentioned on the previous slide, and I purposely avoided that fact because you don't need to know them. And likewise, looking at this slide, I noticed there are some specific pathways leading from the cortex to a variety of these different pathways, and I don't want you to know any of this as well. It's not critical. So for slide number 11, the one we're looking at right now, there really is nothing here you need to worry about. I mentioned the thalamus just a moment ago. The thalamus is located in the diencephalon, and you'll remember that the diencephalon is technically part of the forebrain. It develops part of the prosencephalon, and it's posterior to the telencephalon, which ultimately becomes the cerebrum. So the diencephalon is going to play a role in routing to and from the cerebrum, and the thalamus is going to play a very, very important role in this case. I see the thalamus is illustrated here in green, and its job is to primarily route sensory impulses to various places in the cerebral cortex. And it's going to exercise some degree of judgment, if you will, as to whether those impulses pass to those areas of the cerebral cortex or not. I did mention previously that almost all sensory input gets routed to the thalamus. The only sensory input that does not is the sense of smell, what we're going to call olfactory reception. But fact of the matter is the thalamus, where it says processes and passes, it once again interprets what's coming in, and that allows some of that to pass to other cortical areas. And the thalamus also moderates to some degree some motor output as well. So functionally, it's a pretty darn important part of the brain. Let's take a look at the hypothalamus. The name implies below the thalamus, and I see it's illustrated here as this orange region. And technically, the hypothalamus is right smack dab next to the portion of the brain that we call the midbrain, which in turn connects directly to the brain stem. Now, it wasn't too long ago that I mentioned the brain stem was the site of vital functions. They're sometimes referred to as more primitive functions simply from the standpoint that vertebrates who have complicated central nervous systems, this is where things like heart rate, blood pressure, respiratory rate, etc. are controlled. So what we will see in our brains is that this region of our diencephalon, the hypothalamus, is going to be critical in some of that respect. I see the hypothalamus is going to be involved in controlling cardiovascular and vasomotor centers of the medulla oblongata. It's going to regulate body temperature. It's going to coordinate night-day cycles of activity. It's going to secrete hormones. It's going to control feeding reflexes such as licking and swallowing. And all of the things I just listed are not higher order functions. These are not functions we would associate with the cerebrum. These are vital functions. These are functions that are more critical to maintaining life. So now we come to the brainstem. The brainstem technically is both a combination of the rhombencephalon and the mesencephalon, which was also commonly called the midbrain. Well, let's take a look at the midbrain first. Remember I said that the midbrain is smack dab up against the hypothalamus and the thalamus region of the diencephalon. Well, the midbrain contains some really, really important, I would call them switching and integration centers. One is called the superior colliculus, where you get visual input from the thalamus. Remember, the thalamus brings in a whole bunch of sensory information, but at this point, we're going to process that visual information and we're going to convert it into motor movements of the head and eye. And those are coordinated movements in response to what we see. Obviously, there are going to be pretty darn critical tracks connecting the superior colliculus with the visual centers found in the occipital cortex. Then we've got the inferior colliculus, and the inferior colliculus is going to do the same thing except with auditory input. And that's going to regulate motor outputs as far as the head and trunk in response to those. Because obviously, in any event, when we're looking at something or we're hearing something, we generally are going to react with some degree of gross movement, whether that's eyes and eye focusing or its movement of the body with respect to that sensory input. And once again, I will remind you that midbrain constitutes the developmental endpoint of what started as the mesencephalon way back as a primary vesicle. Then we come to the pons and the medulla oblongata, which together constitute the endpoint of development of the rhombencephalon, which was one of our primary vesicles. The pons, as illustrated there, is kind of tucked on the ventral surface of the appears to be the medulla, and it links the cerebellum to other brain regions, and it regulates shared functions with the medulla oblongata. In other words, a lot of the things that are covered as far as vital processes, heart rate, respiratory rate, blood pressure, all those sorts of things are going to be regulated to some degree by the pons. And the medulla oblongata is where all of these, and it's a lot of them, a whole bunch of motor neurons coming out of the cerebral cortex, coming out of the thalamus, all running into these major tracts that are going to be heading out into the peripheral nervous system, are going to be passing through the medulla oblongata. At that point, something called decussation takes place. These major motor tracts are commonly referred to as pyramids. I don't know why, it's just what they're called. And they crisscross. The motor tracts from the right side cross over to the left side, and the motor tracts from the left side cross over to the right side. That's called decussation. So if you ever run into that term, don't let it cause, a seizure. Decussation means crisscrossing. And it says here, motor tracts from the cerebral cortex cross, right to the left, left to the right. It's what we call contralateral output. And the medulla oblongata is also where we're going to find our autonomic centers. Now, the autonomic centers constitute part of what we call the autonomic nervous system. That's the part of our nervous system that controls involuntary things, like the vital functions that are alluded to on our next to the last bullet. That would be heart rate, respiratory rate, blood pressure, that sort of thing. So basically, the medulla oblongata is extremely vital in terms of what it controls. It is necessary to life. It doesn't deal with any of the higher order things that we see in the cerebrum. And the pons is going to help out in terms of integrating and regulating that between the cerebrum and the medulla oblongata. And one other thing I note under the pons, and it's an area of the brain that we haven't talked about yet, but we will in a minute, and that's the cerebellum, which is going to have an awful lot to do with motor coordination and balance and that sort of thing. And so all of that, the higher order stuff, the motor stuff, not to mention the vital functions, all find themselves wrapped up in their exit from the brain at the level of the medulla oblongata. And that's where we send all this information from the brain to the spinal cord and out into the peripheral nervous system. Last but not least, let's talk about the cerebellum. Cerebellum is technically developed from the rhombencephalon, so it's more closely embryonically aligned with the pons and the medulla oblongata. Functionally, in a nutshell, the cerebellum is all about balance. It monitors proprioceptive information. That's your monitoring of your posture, the position of your muscles, the positioning of your bones, processes all kinds of other things that can contribute to keeping yourself upright, keeping yourself balanced, and keeping yourself muscularly adjusted to what's going on at any given moment. It's an extremely critical and important structure. Now, if you take a look at the illustration there, it is not a good illustration. It's kind of an old-timey pen and ink drawing, and as a consequence, probably didn't cost them anything to put them in the book. But you might be better served if you went ahead and Googled cerebellum, clicked on images. You probably have had some exposure to it in lab as well. Anatomically, the cerebellum has kind of the same organization as the cerebrum. It consists of two hemispheres. This time they're joined by some region called the vermis. It consists of an anterior and a posterior lobe, and the outer surface of the cerebellum features wrinkle-like folds called folia, and those are technically gray matter. That's where we're going to find unmyelinated neurons. And within is branched-like structures referred to as arborvitae, which technically is a Latin name for a shrub. So you've got these branched-like structures of white matter within the folia of gray matter, these two lobes. Well, let's take a look at the spinal cord. In cross-section, and you've probably seen this in laboratory, we see that the spinal cord is a composition of both gray matter areas and white matter areas. The white matter areas are referred to as columns, and those columns are major tracts of myelinated neurons that are running upwards and downwards through the spinal cord, ultimately connecting to the peripheral nervous system. Now, in the very center of the spinal cord, you see the central canal, because it is essentially a hollow organ, and that central canal will connect to the openings or the cavities of the brain, which are called ventricles. And we'll talk more about those when we talk about cerebral spinal fluid. But you will also see gray matter there. Now, the gray matter consists of horns that are described as anterior horns that are projecting forward, posterior horns that are projecting backward, and there are some smaller lateral horns that are projecting to the left and to the right. So what's going to be happening is there is some association, integration going on in the spinal cord. That's where, for example, reflexes are going to be routed in and routed out, bypassing the brain in the process. The major sensory information traveling upwards, ascending to the brain, and descending to our motor and glandular effectors are going to be traveling through the lateral columns, which I said are major myelinated tracts connecting the brain to the peripheral nervous system ultimately. In this slide on gross spinal cord anatomy, you will definitely need to know there are 31 pairs of spinal nerves. You will need to know what constitutes the cervical enlargement and what it controls, what is the lumbar enlargement and what it controls, what is specifically the conus medullaris, the cauda equine, the filum terminale, and the central canal. Everything on this slide is going to be fair game on exam number 4. Much of what is contained on this slide rehearses what we already know about the construction of the spinal cord, at least in cross-section. I notice also we are documenting the different regions in terms of the nerves and the changes in the spinal cord as you progress downwards in terms of cross-sectional structure. There's just a few things on this slide that we hadn't mentioned before. Some of them are definitely going to be necessary for the purposes of exam studying, and 2 others are going to be less important. But the 2 that are less important, you've probably already documented in lab. So I'll mention those first: the posterior median sulcus and the anterior median sulcus. These kind of like grooves in the spinal cord, both on the posterior aspect of the spinal cord and the anterior aspect of the spinal cord. But probably the more important issue here, as far as exam is concerned, are spinal nerves and how they connect to the spinal cord. So spinal nerves constitute the very first portion of the peripheral nervous system coming off of the spinal cord portion of the central nervous system. And these spinal nerves contain both motor and sensory neurons. The motor neurons, you remember, are going to be carrying impulses away from the spinal cord towards effectors, and sensory neurons are carrying them in. Sensory neurons constitute afferent neurons, and motor neurons constitute efferent neurons. But the thing is when an individual spinal nerve connects to the spinal cord, it does so in kind of a pronged fashion. When a spinal nerve approaches the spinal cord, it's going to split. There's one part going to be attached to the spinal cord dorsally, called a dorsal root, and another portion connecting to the ventral portion of the spinal cord called the ventral root. And so you will see that on the top illustration, the cross-sectional view of the spinal cord above. You'll see the yellow spinal nerve, two portions, one called the dorsal root, consisting of the sensory aspect, and the ventral root, consisting of the motor aspect. So in a nutshell, all sensory input is entering the spinal cord through the dorsal root, and all motor output is exiting the spinal cord through the ventral root, both ultimately becoming bound together into the individual peripheral nerve that's going to head its way out into the peripheral nervous system to innervate whatever glands or muscles or other neurons it's headed towards. There's one more observation I want to make about the difference between the dorsal root and the ventral root. The dorsal root, remember, is going to be afferent. It carries sensory information into the spinal cord. The ventral root, on the other hand, is exclusively motor axons and is carrying information away from. It's efferent in nature. But I want to notice there is a ganglion associated with the dorsal root. There are no ganglia associated with the ventral roots of our peripheral nerves. The fact of the matter is these sensory neurons, their cell bodies are located in the dorsal root ganglion, and their axons are going to extend from that dorsal root into the spinal cord. Let's take a closer look at the internal connections and the specific clusters of association neurons within the gray matter of the spinal cord. I'm going to focus on the right side of the illustration where I see the dorsal root with its ganglion entering the dorsal aspect of the spinal cord, and I see within the gray matter, I see the posterior horn, I see the somatic region there consisting of somatic sensory nuclei. These are going to be neurons that are going to be receiving information from the extremities of the body, mostly positions of muscles and bones and things like that. And then I also see a cluster of nuclei referred to as the visceral somatic nuclei, and that's where I'm going to find neurons that are going to be involved in receiving information from the various organs and other internal structures in the body as well. All of this is going to get processed within that gray matter. And then if I shift my attention to the anterior aspect of the spinal cord, where I see the ventral root exiting the spinal cord, I see a rather large pink area there isolated and indicated as somatic nuclei. And those somatic nuclei's job is to basically deal with efferent products of motor impulses going to be traveling out through the ventral root. So once again, all this integration is possible within the spinal cord. I should mention that it doesn't necessarily involve the brain at this point. It is quite possible for information to travel into the spinal cord and be processed and be routed out through a ventral root without necessarily having to be processed by the brain itself. Generally, those sorts of interactions, sensory in, motor out, are called reflexes. And so we have all kinds of things happening on a daily basis where sensory information is collected from our somatic regions, it's collected from our visceral regions, It is routed into the spinal cord, is processed, and is routed out, and I have some kind of result. The central nervous system is surrounded by a triple layered series of connective tissue layers called the meninges. The meninges consist of a very tough outer layer called the dura mater, an area called the arachnoid mater, which also contributes to a space immediately beneath it called the subarachnoid space, and beneath that, the pia mater, which is a very light, thin, much less strong layer where the meninges encounter the actual brain or spinal cord. So let's take a look at the cross-sectional view here, and I notice that most of the business we're going to be learning about concerns the arachnoid mater. Now, arachnoid, that word implies spider-like, so I'm thinking of spider-like openings that occur underneath of the arachnoid mater, that place called the subarachnoid space. That contains this fluid that we're going to call CSF. Stands for cerebral spinal fluid. We'll talk more about that in a minute. That cerebral spinal fluid is flowing all throughout this layer called the subarachnoid space. But the cerebral spinal fluid also needs to have some avenue of getting into the bloodstream, and the bloodstream is going to resupply it. So fact of the matter is, how do I get used CSF from the subarachnoid space into my bloodstream? Well, where you see the dura mater, there are places called venous sinuses. So there are these openings where veins empty, and those venous sinuses are places where we can actually move materials from the cerebral spinal fluid into the bloodstream at that point. There are things called arachnoid villi. And if you take a look at the picture, they look like little club-like structures that seem to be extending from the arachnoid mater, specifically extending from the subarachnoid space into that area of the dura mater, which we're going to call the venous sinus. And there I've got venous blood, and that venous blood is going to be receiving materials moving from the arachnoid villus into the venous circulation. So the meninges are not just limited to the brain. Most times we think about that because we've heard of meningitis, which is an inflammation of the lining of the brain, and we generally associate that with the brain. But the fact of the matter is these meninges are found all throughout the central nervous system, and so therefore we can take a look at the spinal meninges. And the spinal meninges are not that much different from what we see in the brain. I see the same layers, dura mater, I see arachnoid mater, I see a subarachnoid space, and I see pia mater. And so the same layers exist, and they're all going to give me the same basic benefit, which we'll see in a slide or two. Here's where we get a little bit of a clue at what cerebral spinal fluid does for us. Cerebral spinal fluid is located within the meninges, encompassing the central nervous system, and also found within the ventricles of the brain and within the central canal of the spinal cord. And as a rule, cerebral spinal fluid is under some amount of fluid pressure. If you get a leak in the meninges, the cerebral spinal fluid will leak out. It functions as a shock absorber. So if you get a sharp blow to the head or a sharp blow to the spine, the protective nature of essentially a fluid-filled shock absorber is going to prevent damage in those particular sensitive areas of your central nervous system. It's also going to be responsible for diffusing gases and nutrients and wastes and chemical messengers and sorts of things back and forth from your central nervous system. You can kind of think of it as an accessory system for delivery and export of these materials to and from those cells. Now, one last point about this particular slide. We note there's something called the epidural space. Most of y'all have heard of epidurals as an injection that is made generally of some kind of medication into the spine. and it's the epidural space where that injection is made. It's kind of a delicate thing to do, but the fact of the matter is, if the patient bends in a certain way, you can rather easily access this particular region, the epidural space, which exists between the vertebrae and the dura mater of the spinal cord. Well, here's where we get to learn a little bit more about cerebrospinal fluid. I've seen cerebrospinal fluid described as an ultrafiltrate of blood plasma. In other words, it's chemically similar to blood plasma, but it's highly filtered. So it's going to be generated by our circulatory system. Take a look at our slide. It is produced by something called the choroid plexus. Now, to understand what the choroid plexus is, I need to talk a little bit more about the fact that our brain and the spinal cord are hollow structures. I've alluded to the fact that the brain has openings in it called ventricles. There are technically 4 ventricles in the brain, 1 in each of the right and left cerebral hemispheres. There's one located down near the diencephalon, right near where the thalamus is, in the middle of your brain. And then there's another one associated with the brain stem. Those ventricles are all interconnected, and they ultimately connect to the central canal of the spinal cord. Now, these spaces, these ventricles, are lined with a network of blood vessels referred to as the choroid plexus. The choroid plexus is a network of blood vessels that are going to be responsible for generating cerebrospinal fluid. That cerebrospinal fluid enters the ventricles and bathes the brain and spinal cord with this fluid. And by virtue of our body movements, it's constantly circulating, and it's constantly being produced by the choroid plexus, and it's constantly being reabsorbed by the arachnoid villi in combination with the venous space in the dura mater. Our brain, and more specifically the cells of our central nervous system, are very sensitive to some things, chemical compounds, toxins, that don't belong. And for this reason, we have a specialization of the intersection between our bloodstream and our brain called the blood-brain barrier. Don't think of it as a single wall between the outside of our brain and the inside. What it is, it's a barrier that exists between our nervous tissue and every single teeny tiny blood vessel that carries blood into that nervous tissue. So, first off, imagine capillaries. Capillaries are the smallest of our blood vessels. And those smallest of our blood vessels are lined by a very, very thin layer of what we're going to call endothelial cells. Endothelial cells do not really provide a tremendous barrier to things crossing. Oxygen can cross, carbon dioxide can cross, many, many other molecules can cross, either cross from the blood into the brain or from the brain back into the blood. But in addition to these epithelial cells, there is a layer of connective tissue surrounding all of these capillaries, and there's also these cells we had mentioned once upon a time called astrocytes. And you remember that astrocytes are a type of glial cell, and they're responsible for delivering nutrients to our neurons. Our neurons do not partake of materials from the bloodstream directly. It's the astrocytes that do it for them. So what happens is the combination of endothelial cells, connective tissue, and astrocytes makes certain that only the molecules I need, food molecules, various hormones, other supportive molecules, oxygen, those get to my neurons. Things that I don't want, chemical compounds that are toxic, drug metabolites, things like that, don't get there. So I noticed that my slide tells me that the blood-brain barrier maintains a stable environment for the central nervous system. It's going to involve selective permeability. What that means is we get to regulate what passes through. I'm going to let nutrients in, I'm going to let wastes and toxins out. It's going to be ineffective against fats and gases, but that's fine. I just want to make sure that my neurons get only what they need. And in doing so, I'm also protecting my central nervous system from things that I don't need, I don't want, things that could be potentially dangerous. Okay, we've talked about the brain. We talked about the embryonic origins of the brain, talked about anatomy of the brain, talked about spinal cord, talked a bit about the anatomy of the spinal cord. Now it's time to start talking about nerves. We've not talked about the composition of nerves, and they dominate our nervous system. They dominate our peripheral nervous system, obviously. This is how we carry sensory information into our central nervous system, and it's how we carry motor information out. What's a nerve? A nerve is like a big highway of multiple axons. You can think of a nerve as almost like a telephone cable. If you've ever taken a look at a telephone cable, you realize it's nothing more than a big insulated cable full of teeny tiny little wires. And nerves are much the same way. A nerve consists of something called an epineuron, which is a layer of connective tissue that entirely surrounds the nerve. Within the nerve, there's going to be bundles of axons, and those bundles are called fascicles. And those fascicles are surrounded by its own layer of connective tissue called a perineurium. And then finally, each individual axon is surrounded by a layer of connective tissue called the endoneurium. So these nerves can be classified into one of two general groups: cranial nerves or spinal nerves. And we already mentioned they can also be classified on the basis of the direction of transmission. Sensory nerves, consisting of sensory neurons, carrying information into the central nervous system, hence also called afferent neurons, and motor nerves, consisting of motor neurons, carrying information away from the central nervous system, that is, efferent nerves, or efferent neurons. And then finally, there are some nerves that are classified as mixed, and they can carry both motor and sensory axons. Now, when I say axons, you will remember that our cell bodies of our neurons are located in swellings in the peripheral nervous system called ganglia, or found in specific areas within the brain called nuclei. Regardless, your axons, which are significantly long parts of these neurons, need to be bundled into these major pathways, these major tracks, so I can carry these impulses to and from the central nervous system over significantly long distances. So technically, that's what a nerve is. It is a bundle of axons. Cell bodies are either located in ganglia in the peripheral nervous system or in nuclei in the central nervous system. So what's up with these cranial nerves? Well, you learned about cranial nerves in laboratory, I'm sure, and you identified them. So once again, I'm just going to say you probably should rely on what you learned in the laboratory. There are 12 pairs of cranial nerves. Cranial nerves, as a rule, issue from the brain or the brain stem. Cranial nerves can be sensory or motor or mixed. And the first two pair come from the brain itself. That would be the olfactory and the optic nerves, and all the rest of them issue from the brain stem. So, Dr. Knaus, I guess I need to know the names of the cranial nerves. I guess I need to know the numbers of the cranial nerves. And I probably ought to know whether they're motor or they're sensory or they're both. And it'd probably be a really good idea, Dr. Knaus, if I knew what sorts of information the cranial nerve was carrying. I think that's a pretty astute observation, because I can guarantee you there's going to be multiple questions on this exam that will probably deal with these cranial nerves. So you'll want to have that under your belt and ready. To that end, this is an important slide, and I'm sure it reproduces information that's found in a table in the book, or you could certainly Google it and come up with all kinds of versions of the same thing. You might remember that earlier in this presentation, I said, well, you're probably going to need to know the names of the spinal nerves. At least you need to know how many there are. And you need to know that they're grouped into a variety of regions: the cervical spinal nerves, the thoracic spinal nerves, the lumbar spinal nerves, the sacral spinal nerves, and a coccygeal spinal nerve. So make sure you know the numbers, and make sure you know the names, and make sure you know where they're located. And one of the common designated ways of referring to these spinal nerves is to use the letter of the region and then add a number indicating the particular spinal nerve you're talking about. So, for example, cervical nerves are all indicated with a C, 1, 2 through 8. The thoracic nerves identified with a T, 1 through 12. Lumbar nerves, L, 1 through 5, etc. Finally, some imbalances or disorders you want to be aware of. First off, the chickenpox virus, which causes chickenpox, standard childhood disease, can actually remain dormant within nerves, recurring periodically as something that is called shingles. Fainting, also known as syncope, is a result of a decreased blood flow to the brain. Many people will become faint if they stand up very quickly and they already have low blood pressure. Standing up can all of a sudden cause a significant drop in blood flow to the brain, causing a fainting event. Stroke, because of lack of oxygen to brain cells. Meningitis, inflammation of meninges can either be bacterial or viral in origin. Spina bifida, the incomplete closing of the backbone and membranes about the spinal cord. Each of these can certainly be Googled, and I strongly suggest that you know these because there's probably going to be a question or two on exam number 4.